Okay, here we go. Chapter 9, the cell cycle. The ability of organisms to produce more of their own kind best distinguishes living things from non-living matter. Continuity of life is based on the reproduction of cells or cell division. So this is tracking cells' chromosomes over the course of cell division. In unicellular organisms, division of one cell reproduces the entire organism. Cell division enables multicellular eukaryotes to develop from a single cell, and once fully grown to renew, repair, or replace cells as needed. Cell division is an integral part of the cell cycle, the life of a cell from formation to its own division. So here are 3 functions of cell division: reproduction, growth and development, and tissue renewal. Most cell division results in the distribution of identical genetic material, or DNA, to 2 daughter cells. So this, for example, would be mitosis. DNA is passed from one generation of cells to the next with remarkable fidelity. All the DNA in a cell constitutes the cell's genome. A genome can consist of a single DNA molecule, such as that in prokaryotic cells or bacteria, or a number of DNA molecules, which is common in eukaryotic cells, like our own cells, where we have 46 chromosomes, 23 pairs. DNA molecules in a cell are packaged into chromosomes. This is what eukaryotic chromosomes would look like under the microscope. These are highly condensed molecules. Eukaryotic chromosomes consist of chromatin, which is a complex of DNA and protein. And the protein that's associated with the DNA, those are called histones, and the DNA wraps around the histones. Most eukaryotic species has a characteristic number of chromosomes in each cell nucleus. Just like in our cells, we have 46 chromosomes in our body cells. That excludes our sex cells, our egg and sperm, which only have 23 chromosomes. Somatic cells, or body cells, these are your non-reproductive cells, have 2 sets of chromosomes. In humans, again, that's 46. In the gametes, the reproductive cells, the sperm and the eggs, have 1 set of chromosomes. In humans, that's 23. In preparation for cell division, DNA is replicated, and the chromosomes condense. Each duplicated chromosome has 2 sister chromatids, which are joined identical copies of the original chromosome. The centromere is the part where the 2 sister chromatids are most closely associated to each other. We could also say that that's where the sister chromatids are attached. During cell division, the 2 sister chromatids of each duplicated chromosome separate and move into 2 nuclei. Once separate, the chromatids are called chromosomes. So here we have a chromosome. It is duplicated before cell division. So here we have a duplicated chromosome with 2 sister chromatids that are connected at the centromere. And then after cell division, we have the separation of those 2 sister chromatids into separate cells. Eukaryotic cell division consists of mitosis, which is the division of the eukaryotic nucleus, and cytokinesis, which is the division of the cytoplasm and all of the organelles that are found within the cytoplasm. Gametes are produced by a variation of cell division called meiosis. Meiosis yields non-identical daughter cells that have only 1 set of chromosomes, half as many as the parent cell. And meiosis also leads to genetic variation in those daughter cells. The cell cycle consists of the mitotic phase, which includes mitosis, the division of the eukaryotic cell nucleus, and cytokinesis, which is the division of the cytoplasm. During interphase, interphase is the phase that comes before mitosis, before the division of the nucleus. So during interphase, what happens is the DNA gets copied or replicated, organelles get replicated, and cell growth occurs. This is about 90% of the cell cycle, and it consists of the G1 phase, the first gap, the S phase, the synthesis phase, where DNA gets replicated, and G2 phase, the second gap, where you have more growth of the cell. Okay, so here is your little pie chart, which shows the cell cycle. So notice that the majority of the pie chart is interphase, so G1, S, and G2. And then you have this small little portion that's mitosis and cytokinesis. Mitosis is conventionally divided into 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. Cytokinesis overlaps the latter stages of mitosis. Okay, so here we have interphase. You can see that the chromosomes aren't very condensed here. They're sort of just this long string-like material. You can see that the nuclear envelope is still present. You can see your centrosomes here with centriole pairs, and you can see that the nucleolus is still visible. And then during prophase, you can start to see the production of your early mitotic spindle. Your nuclear envelope is going to start to disappear. This is an aster. This is where your centrosomes have little spindle fibers starting to come out of it. And then you can see that the chromosomes have been condensed. Oops, wasn't done with that slide. Prometaphase, you can see that you have microtubules associated with the chromosomes. So the kinetochore microtubules are the ones that are going to attach to the centromere of the chromosomes. And then the non-kinetochore microtubules, these are going to be the ones that are going to help elongate the cell. You can see that the nuclear envelope has pretty much disappeared, and you can see that the centrosomes have moved to opposite poles. And then you have metaphase, where the duplicated chromosomes line up individually at the metaphase plate or the equator. And then anaphase, the kinetochore microtubules start to shorten and pull the sister chromatids apart. And then telophase and cytokinesis. In animal cells, you have a cleavage furrow forming where your cell membrane pinches in. Your nucleolus starts to reform. Your spindle has disappeared, and your nuclear envelope starts to reform. This is all the same. The mitotic spindle is a structure made of microtubules and associated proteins. It controls chromosome movement during mitosis. In animal cells, assembly of spindle microtubules begins in the centrosome, the microtubule organizing center. The centrosome replicates during interphase, forming 2 centrosomes that migrate to opposite ends of the cell during prophase and prometaphase. An aster, radial array of short microtubules, extends from each centrosome. The spindle includes the centrosomes, the spindle microtubules, and the asters. During prometaphase, some spindle microtubules attach to the kinetochores of the chromosomes and begin to move the chromosomes. So these are the ones that are going to pull the sister chromatids apart. Kinetochores are protein complexes that assemble on sections of DNA at the centromeres. Remember, the centromere is the middle section of the chromosome that holds the 2 sister chromatids together. At metaphase, the centromeres of all the chromosomes are at the metaphase plate, an imaginary structure at the midway point between the spindle's 2 poles. And again, here's another picture of that. Your kinetochore microtubules that are attaching to the centromeres, or the kinetochores of the chromosomes, and the overlapping non-kinetochore microtubules that will function to elongate the cell. In anaphase, sister chromatids separate and move along the kinetochore microtubules toward opposite ends of the cell. The microtubules shorten by depolymerizing at their kinetochore ends. Chromosomes are also reeled in by motor proteins at spindle poles, and microtubules depolymerize or break down after they pass by the motor proteins. So this is just a picture of what's happening when the kinetochores are pulling the sister chromatids apart. They start to break down. Non-kinetochore microtubules from opposite poles overlap and push against each other, elongating the cell. At the end of anaphase, duplicate groups of chromosomes have arrived at opposite ends of the elongated parent cell. Cytokinesis begins during anaphase or telophase, and the spindle eventually disassembles. In animal cells, you have the formation of the cleavage furrow, where basically your cell membrane starts to pinch in, which then allows the cell to basically become 2, break into 2. In plant cells, you have the formation of a cell plate, which forms from vesicles that butt off of the Golgi, and then that cell plate develops into the cell wall of the plant cell. Here's what the phases would look like under the microscope. Here, prophase, you can see that the chromosomes aren't very condensed, as opposed to prometaphase, where you can see them really super condensed here. And then metaphase, they're lining up in the middle. Anaphase, they're starting to be pulled apart. And telophase, this is a cell plate right here. So prophase, they start to condense. Prometaphase, they're really condensed. Metaphase, they're found in the middle. Anaphase, they start to pull apart. Telophase, formation of a cell plate. Prokaryotes reproduce by binary fission, which is a very simple process where basically the DNA replicates, the bacterial chromosome replicates, and the cell just splits in half. So super fast, super simple. Your two cells are identical to the original cell. Since prokaryotes evolved before eukaryotes, mitosis probably evolved before, evolved from binary fission. So binary fission came first, and then mitosis probably evolved from that. The frequency of cell division varies with the type of cell. These differences result from regulation at the molecular level. Cancer cells manage to escape the usual controls on the cell cycle. So cancer is just basically there's no cell cycle regulation. The cell just divides out of control, eventually becoming a tumor. We don't have to worry about this. The sequential events of the cell cycle are directed by a distinct cell cycle control system, which is similar to a timing device of a washing machine. The cell cycle control system is regulated by both internal and external controls. The clock has specific checkpoints where the cell cycle stops until a go-ahead signal is received. So there's the G1 checkpoint. If the cell gets past the G1 checkpoint, more likely than not, it will finish dividing. There's also the G2 checkpoint or the M checkpoint. So basically, if there are any problems during interphase where the cell is not ready to divide, it will go out of the cell cycle and enter the G0 phase. So for many cells, the G1 checkpoint seems to be the most important. If a cell receives a go-ahead signal at the G1 checkpoint, it will usually complete the S, G2, and M phases and divide. If the cell does not receive the go-ahead signal, it will exit the cycle, switching to a non-dividing state called the G0 phase. So without go-ahead signal, the cell enters G0. With the go-ahead signal, the cell will continue the cell cycle. So for example, at the M checkpoint, without full chromosome attachment, a stop signal is received. So if the microtubules are not all attached to the chromosomes, this is going to be a problem. When the cell divides, you could end up with too many chromosomes in one cell. So that would be a problem. So the cell would stop. It would go into the G0 phase until the problem could be repaired. The cell cycle is regulated by a set of regulatory proteins and protein complexes, including kinases and proteins called cyclins. An example of an internal signal occurs at the M phase checkpoint. In this case, anaphase does not begin if any kinetochores remain unattached to spindle microtubules. Attachment of all the kinetochores activates a regulatory complex, which then activates the enzyme separase, which then causes the sister chromatids to separate at their centromeres. Don't worry about that. Actually, we will talk about that. Some external signals are growth factors, proteins released by certain cells that stimulate other cells to divide. For example, platelet-derived growth factor stimulates the division of human fibroblast cells in culture. So here we have the effect of platelet-derived growth factor. A sample of human connective tissue is cut up into small pieces. Enzymes digest the extracellular matrix, resulting in a suspension of free fibroblasts. The cells are then transferred to culture vessels, and you can see that without PDGF, the growth factor, you don't have much growth of the cells, as opposed to with that growth factor, you have the growth of many of those cells. Another example of external signals is density-dependent inhibition, in which crowded cells stop dividing. So this is important because if you have crowded cells continue to dividing, that basically is cancer. So you would want to have this inhibition to prevent the growth of cancerous cells or tumors. Most animal cells also exhibit anchorage dependence, in which they must be attached to a substratum in order to divide. This is also important because with cancer, metastatic cancer, you have cells, cancerous cells that are becoming unattached to other cells and moving to other parts of your body, and that's causing cancerous cells to develop in other tissues and organs. Cancer cells exhibit neither density-dependent inhibition nor anchorage dependence. So anchorage-dependent cells require a surface for division. Density-dependent inhibition, cells form a single layer. Density-dependent inhibition, cells divide to fill a gap and then stop. So this would be normal cells, where they would stop dividing, versus cancerous cells, where they would just continue to divide, even though that's more than a single layer. Cancer cells do not respond to signals that normally regulate the cell cycle. Cancer cells may not need growth factors to grow and divide. They may make their own growth factor. They may convey a growth factor signal without the presence of the growth factor. They may have an abnormal cell cycle control system. A normal cell is converted to a cancerous cell by the process of transformation. Cancer cells that are not eliminated by the immune system form tumors, masses of abnormal cells within otherwise normal tissues. So your immune system is supposed to target and kill cancerous cells, but the problem with a cancerous cell is that it looks like a normal cell. Your immune system cells and antibodies and your B cells and your T cells, they recognize foreign material, and that foreign material is basically differences in glycolipids and glycoproteins on the surface of foreign cells. So any foreign material, any foreign protein, any foreign carbohydrate, but cancerous cells, they look like your cells because they come from your cells. So it's very hard for the immune system to recognize and destroy cancerous cells. If abnormal cells remain only at the original site, the lump is called a benign tumor. Malignant tumors invade surrounding tissues and can metastasize or move to other parts of the body. So here we have the growth and movement of a tumor or of cancerous cells in breast cancer. So step 1, a tumor grows from a single cancerous cell. Cancer cells invade neighboring tissues. Cancer cells spread through lymph and blood vessels to other parts of the body. And a small percentage of cancer cells may metastasize to another part of the body. Recent advances in understanding the cell cycle and cell cycle signaling have led to advances in cancer treatment. Medical treatments for cancer are becoming more personalized to an individual patient's tumor. One of the biggest lessons in cancer research is how complex cancer is.